Hello, everyone, and welcome to today's session on bioinformatics for precision oncology. Let's go ahead and get started with today's session. Today, we're going to be talking about somatic and germline mutations, as well as the mapping and detection of variants. We'll also be doing some interpretation to decipher significant versus insignificant mutations. Before we do that, let's talk a little bit about course progress. So here's the graphic of the course progress since last Thursday. And we can see that some of you have made quite a bit of progress since last week.
Yeah, Joe, we can hear you. Everything's good.
Oh, you can hear me? Okay.
Yeah. I don't know. Mohit maybe has a problem, but can everybody hear Joe? If you can't hear him, just maybe put it in chat.
I can hear you, Joe. You're audible.
Okay. So since last week, we've got quite a bit of progress. It looks like Asma, Nicholas, and Dahlia have done quite a bit of work. I guess I could read all of these, but it looks like a lot of you have kind of done quite a bit on the education side. And I think that as we begin to go through the course and start running some pipelines, you'll see your points picking up on the server side. So we're going to do a little bit of a review of some of the things that we talked about last week. And then I'm going to have a couple of quiz questions for you before I get started with today's session. So let's go over some of the hallmarks of cancer that we've identified last time. These are distinctive and complementary capabilities that enable tumor growth, as well as metastatic dissemination. So these have evolved over the past decade, and they now include sustaining proliferative signaling, evading growth suppressors, the activation and invasion of metastases, the ability to enable replicative immortality, inducing angiogenesis, and then resisting cell death. There are additional 4 that we can talk about, and that would be deregulating cellular energetics, avoiding the host immune destruction, promoting tumor inflammation, and then genome instability and mutation. So let's go through these again. This is sustaining proliferative signaling. And then we have evading growth suppressors, activating the invasion and metastases, enabling replicative immortality, inducing angiogenesis, and then resisting cell death. Next generation sequencing technology captures DNA sequences and generates a digital data using several different steps. First, RNA that was extracted from cells is going to be converted to cDNA that's going to be sheared and placed into a flow cell. Inside the flow cell, these cDNA fragments are going to be amplified using a process called bridge PCR amplification. And then the flow cell is going to be inserted into the sequencer. This machine uses image analysis to capture each letter in the flow cell fragments by analyzing visual patterns and then converting those into a sequence of letters. Typically, NGS reads are going to be between 30 and 300 base pairs long, and they consist of a series of letters of T, C, G, and A, and those are the nucleotides. Next generation sequencing technology allows for advanced studies of gene expression because it captures a snapshot of the whole transcriptome rather than a predetermined subset of genes. And this is what was previously possible with reverse transcription PCR and microRNA technologies. Whole transcriptome sequencing provides a comprehensive view of cellular transcriptional profiles at any given biological moment And what it does is it greatly enhances the power of RNA discovery methods. In order to study this data, we have to run processing pipelines that turn raw reads into structured data that's ready for analysis. So why study genomics? Genomics is an essential subfield of bioinformatics, and it's a major force in expanding human knowledge of genetic associations with disease as well as other traits. It's an interdisciplinary field, and it includes the methods for DNA sequencing as well as for big data analysis of genomic sequences. NGS techniques allow for whole genome sequencing and the analysis of epigenetic factors such as DNA protein interactions and DNA methylation with unprecedented efficiency. Massive databases for biological sequences can be GenBank, which is the National Center for Biotechnology Information, EMBL, which is the European Bioinformatics Institute, as well as SRA, which allow for data-driven research and knowledge discovery. There's large amounts of data from the 1,000 Genomes Project that are publicly available, and they provide anyone with proper analytical skills and resources the opportunity for scientific discovery. While transcriptomes are what we're going to be studying, there are, and as well as the genome, the transcriptomics seeks to find various variation in gene expression through methods such as RNA sequencing, genomic studies, and then genomic-wide genetic variation at the fundamental level of DNA sequences. To understand the genetic associations that are provided by genomic research, we'll talk about how they have great value in medicine, agriculture, ecology, biotechnology, and other industries. I'm going to go over a couple of quiz questions regarding the cell cycle and cancer. And so if you want, go ahead and answer in the chat. So this first one is that normal cell division is prevented by which of the following? Is it proto-oncogenes, oncogenes, cell death, or cell multiplication? So we've got one answer that proto-oncogenes prevent normal cell division. Two for that. So the answer choice for this is actually oncogenes. Let's try another one. P53 activates which of the following? Tumor suppressor genes, DNA repair proteins, the cell cycle, or uncontrolled cell division? So it looks like, is it ZNAB has the correct answer? So P53 activates DNA repair proteins. Damage to which type of genes is the most common cause of cancer? Tumor suppressor genes, oncogenes, proto-oncogenes, or cell death inducing genes? So there's only one correct answer for this. I could argue that two possibly, but the correct answer is going to be damage to tumor suppressor genes. Obviously, so that would be something like P53. So I'll pause now and see if any of you have any questions before I get started with the material. I saw someone said something about the website slowing down earlier. I believe that has a lot to do with people running a ton of pipelines right now. So that should resolve itself. Okay, we can, if you want, I can just kind of give you a brief demonstration of how to do things on the website. And then you're also going to be getting some of this from Elia later on in the session, but I can pop out real quick and kind of show you.
Joe.
Yeah.
Just maybe read out the questions because I don't think everybody has.
Oh, okay. Someone asked, they wanted to see the pipelines on the server, how to run one. And so let me.
Let me do that later. If you can go through your part of the session, and I'll do the pipelines because they're going to be genomic pipelines, so I don't want to confuse them with the RNA-seq pipelines.
Okay, that sounds good. So we'll be going over those in just a little bit. Let's discuss a little bit more about Genomics 1 as a course. So we can see that we have these different sections here where we have a review of DNA variation. Let me pull this up. Okay, so yeah, we're going to be going over the pipelines later on. So anyways, there's a review of DNA variation. So we're going to talk about the DNA code basics, point mutations, rearrangements, insertion, and deletions. We're going to talk about the types of information that are involved in genomics. So those would be whole genome sequencing or whole exome sequencing. We'll also talk about RNA sequencing and targeted sequencing. There are example projects for you to learn about, one having to do with Huntington disease, which we'll discuss today, cancer, tuberculosis, and Ebola. And then we're also going to be discussing the challenges of genomic analysis in sequencing DNA, the alignment of reads, and the detection and significant changes, as well as the interpretation of the results that we're getting. Let's talk a little bit about chromosomes. DNA molecules are tightly packed around proteins that are called histones, and they make structures called chromosomes. So a human being has 23 pairs of chromosomes in every cell, which makes 46 chromosomes in total. Sex chromosomes are what determine gender. So males have XY chromosomes and females have 2 X chromosomes. There are other types of chromosomes and they're called autosomes. The largest chromosome, which is chromosome 1, contains about 8,000 genes. The smallest is significantly smaller, and it's chromosome 21, and it contains about 300 genes. So by now, most of you have seen the structure of DNA. We're going to talk a little bit about how DNA is made up. So genes are made of a chemical that are called DNA, which is short for deoxyribonucleic acid. DNA forms a double helix in solution, and it has 2 long, thin strands that are twisted around each other, like a spiral staircase. The sides and the backbone of the molecule are going to be formed of sugar and phosphate. So sometimes you might see it referenced in literature as a sugar-phosphate backbone. Let's go in and zoom in and kind of look at how these bases form. So the way that these bases are put together are as nitrogenous bases, or you can call them bases for short. There's 4 types of bases: adenine, thymine, guanine, and cytosine. A pairs with T and G pairs with C. And then we know that there's these different type of nucleotides have different classifications. So nucleotides with 2 rings are called purines, and nucleotides with 1 ring are called pyrimidines. One way that you can remember this is if you call these purines. You can remember this as purines or call them pure rings, so that you remember that the purines are the ones with the 2 ring structures, and then those are different. So in DNA, thymine is the pyrimidine that's used to pair with adenine. In RNA, you'll learn later, is uracil is what is used. And the difference between those is that on the 5 position, there's no methyl group here. So if you simply just deleted that CH3 group on position 5, then you would have uracil. There's also a genetic code. So we're talking about translation now, where the mRNA transcript is going to be read by the ribosome, and based on the 3 codon sequence, a specific amino acid is going to be formed. There are 20 standard amino acids that are formed in proteins that are, I guess, of biological origin. So for instance, if you have a TTT, so 3 thymines together, then that would be read by the ribosome as needing to code for phenylalanine. For instance, if you had AGA, so adenine, guanine, adenine, then the codon would be read as arginine, which is another amino acid. There are specific stop codons. So once those bases are read by the ribosome, then translation will stop. Those are going to be TAA, TAG, and TGA. Another thing to note is that translation starts at a very specific codon, which would be ATG, and that codes for methionine. In bacteria, there's a formal group on there, and so formal methionine is how translation starts with those. A point mutation or substitution is a genetic mutation where a single nucleotide base is changed, inserted, or deleted from a sequence of DNA or RNA. Point mutations have a variety of effects on the downstream product of a protein. These consequences are moderately predictable based on the specifics of the mutation. So we have a substitution type of mutation. That's where one base is going to be substituted for another. So we see here that T is substituted in and C is substituted out. There's also insertions. So for instance, adenine being inserted into the base or into the nucleotide structure. And what happens then is there's going to be a frame shift in the codon. And then similarly, a deletion can occur where you take a base out, and then that's going to also cause a frame shift. There's also rearrangements. So in genetics, there's a chromosomal rearrangement, which is a mutation that's a type of chromosome abnormality involving a change in the structure of a native chromosome. Such changes involve several different classes of events, such as deletions, duplications, inversions, and translocations. Insertions occur when a nucleotide is inserted into the sequence, which results in a frame shift. And then frame shifts also occur via deletions. One of the projects that you're going to be learning about in this program is Huntington's disease. This is a germline mutation, and it's an inherited condition where a specific gene on chromosome 4 has an abnormal number of CAG codons. The HTT gene encodes the large Huntington protein, and the additional repeats change the protein structure, resulting in a progressive movement disorder with dementia, as well as behavioral abnormalities. There are different clinical signs of Huntington's disease, and they typically emerge midlife. They have a juvenile onset as well as elderly onset as far as cases that are being seen. The diagnosis of Huntington's disease is based upon the presence of a characteristic motor sign, but the disorder includes intellectual decline as well as psychiatric disturbances. The median age of death for Huntington's disease is about 18 years after the onset. In 2015, the Huntington Consortium published a study of over 1,000 individuals with early and late onset of the disease. They applied targeted sequencing as well as whole genome sequencing. And the researchers discovered important regulation mechanisms that differentiated between those groups and provided insight into other mutations. We can see here that there's one on chromosome 15. And these were associated with slower development of clinical onset. This study, as well as others, are good examples of how genomic studies can help identify mechanisms of disease progression and potential therapeutic interventions leveraging natural mechanisms of disease regulation. Let's talk a little bit about somatic mutations. So these mutations that we're talking about with Huntington's disease are germline, so with the sex cells. Somatic mutations are those for body cells. So if a somatic mutation occurs in a single cell or developing somatic tissue, the cell that is the progenitor of a population of identical mutated cells, all of those which descend from that will be mutated. A population of identical cells derive asexually from one progenitor cell, and it's going to be called a clone. So the cells that come from somatic mutations are going to be called clones. Because the members of a clone tend to stay close to one another during development, an observable outcome of somatic mutation is often a patch of phenotypically mutated cells that are called a mutant sector. The earlier in development the mutation occurs, the larger the mutation sector will be. Mutant sectors can be identified by eye only if their phenotypic contrasts visually with the phenotype of the surrounding wild type cells. So with somatic mutations, you have acquired mutations, and they're the most common cause of cancer. Germline mutations are less common. And then with tumor suppressor genes, these are protective genes. Oncogenes turn a healthy cell into a cancerous cell. And so those are kind of a description of some of the things that we talked about in the quiz earlier. Regarding that part of the material, does anyone have any questions? So we have a question. It says, I always get confused about insertions and deletions that are less than 100 base pairs, which are not copy number variants nor single nucleotide point mutations. What would you call those types of insertion deletions? So ones that are less than 100 but more than one.
So I can maybe answer that.
Okay.
So the differences between copy number variations typically refer to copies of a gene, or for example, a coding portion, which could be an exon. There are indeed many different types of variants I talked about. We'll talk about single nucleotide mutations, which will be one or at least maybe several nucleotides long. But indeed, there are less than 100 base pairs, so 50, 70 different variants. The problem about those is that if you think about a read, right? So depending on the length of your read, that could be easier or more difficult to find. If your reads are longer than 100 base pairs, then you can easily find those because you will have to have at least a substantial portion of the read mapped to the reference genome, giving you the right position of that read. And then in some reads, you will have a missing portion of those reads, right? So let's say if your reads are 300 base pairs long and your deletion is 100, or let's say 50, then you still have enough of that read mapped to elsewhere to be able to identify. The problem is that there's not so much long read sequencing for a significant number of samples oftentimes, to really identify those. And the quality of mapping, so a precise identification of where the read maps on the reference genome, goes down the less reads you have left in a read that are not deleted. So that is the challenge. And so when we talk about these deletions that are less than, let's say more than 50, but less than 100 base pairs long, we're really facing a challenge of accurate mapping and sufficient number of samples to be, to have a statistical confidence that that is indeed a full deletion or insertion, and not just some kind of a combination of sequencing error, low depth, etc. So those are not reported as often and with high enough confidence as the shorter single nucleotide variations, or when we have whole genes that are missing, right? Those are, in a sense, easier to identify because and we will talk about some of those examples today. So I hope that answers your question.
All right. Well, I guess we're going to talk a little bit about data preparation now, if there's no further questions. All right. So we're going to start a little bit with Sanger sequencing, which is a first-generation sequencing method. And then we're going to discuss Illumina. Sanger sequencing is a first-generation DNA sequencing method. Despite the advantages of next-generation sequencing techniques, where throughput is orders of magnitude higher, Sanger sequencing retains an essential place in clinical genomics for at least two specific purposes. First, Sanger sequencing serves as an orthogonal method for confirming sequence variants that are identified by next-generation sequencing. When validating clinical NGS tests, reference materials sequenced by Sanger approaches provide ground truth, which the NGS assay can be benchmarks. These materials may include well-characterized publicly available reagents, such as cell lines studied by the HapMap project, or archival clinical samples that are tested by Sanger methods. So we're going to be discussing the different type of chemistries that occur with Sanger. It'd be impractical to Sanger confirm every type of variant, given the large number of primers, reactions, and interpretations that would be required. However, there may be instances where the veracity of a specific variant is in doubt. For example, called variants that are biologically implausible or otherwise suspected of being spurious. Sanger sequencing is the easiest method to resolve these uncertainties, and therefore it is an invaluable protocol in any clinical genomics laboratory. But as we'll see later, next generation sequencing is much more high throughput. So here's an example of the Sanger sequencing method. So it's also called the classical chain termination method, and it requires a single-stranded DNA template, a DNA primer, DNA polymerase, and then a normal deoxynucleotide triphosphate. And then there's also a specific type of modified molecule called a DDNTP, which is a dideoxynucleotide triphosphate. And this DDNTP molecule is what terminates chain elongation. What this does is it attacks the 3' hydroxyl group that's required for the formation of the phosphodiester bonds between different nucleotides. And so as you can see here on the graphic, what's occurring is that this DDNTP is going to be incorporated at the end of the 3' hydroxyl group here. And it's going to become progressively shorter and shorter. And so by doing that, you're able to then put this through a capillary gel and detect it, and then know which type of nucleotide you have. Now let's contrast that to something such as Illumina. which allows researchers to explore genetic variations like never before, down to single nucleotide resolution. Whether you need to identify specific variants from a focused set of regions or look more broadly for potential causative variations, whole exome sequencing or whole genome sequencing both offer effective solutions. Let's talk a little bit about the differences between whole exome sequencing and whole genome sequencing. So whole exome sequencing is a targeted view of the protein coding regions of the genome. It offers reliable and sensitive detection of coding variants. It's fast and cost effective. However, whole genome sequencing provides a comprehensive view of the genome, and it includes the coding, non-coding, and mitochondrial DNA regions. It's reliable and sensitive as well, but includes the detection of all of the variant types. So those would be SNVs, indels, SVs, and CNVs. It's low cost and fast as well for library preparation. The data for whole exome sequencing is prepared using two approaches, the first of which is array-based, and then the second is going to be in solution. So for array-based capture, microarrays capture single-stranded oligonucleotides with sequences from the human genome to tile the region of interest fixed to the surface. Genomic DNA is sheared to form double-stranded fragments, and these fragments undergo end repair to produce blunt ends on the adapters. And these have universal priming sequences that are going to be added. These fragments are hybridized to oligos in the microarray. Unhybridized fragments are going to be washed away, and then only the desired fragments will be eluted. Those fragments that are eluted are going to be amplified using polymerase chain reaction. And this contrasts to in-solution capture, where the captured genomic regions of interest are used in solution to capture and then pool custom probes. These are going to be synthesized and hybridized in solution to a fragmented genomic DNA sample. These probes are labeled with beads, and they're selectively hybridized to the genomic regions of interest. And then these can be pulled down and washed to clear the excess material. So this is what's happening in a beaker. The beads are then going to be removed, and the genomic fragments can be sequenced. And then those genomic fragments are sequenced for selective DNA sequencing of the genomic region of interest. So here we can see the targeted panel sequencing, and this has between 400 and 400 genes. Whole exome sequencing has around 2,000 genes, and then whole genome sequencing is all of the genes and non-coding DNA. So as we see here, we have high coverage, intermediate coverage, and lower coverage. I'm going to pass the presentation over to Ilya, and he's going to go over some of the practical steps for you to cover. And then I will probably see you again next session.
Thank you, Joe. Okay, let me share the screen once you stop sharing yours. So all of you have been waiting to get started with the practical examples, and so I'm kind of excited to start sharing that process with you. I do want to mention that, just to clarify a couple of things that Joe said, he did a very good overview of some of the introductory material about the mutations, point mutations, single nucleotide variations, and the different types that we expect to find, for example, germline versus somatic. So that's what we're going to be talking about today. He mentioned a few other examples that have been used in previous illustrations: Huntington disease, Ebola, tuberculosis, etc. We're not going to be focused on those examples in this program. This program is going to be dedicated to the cancer examples. And the cancer examples, both of those mutations are important, right? So germline mutations, can anyone think of a germline mutation that is very common in breast cancer? BRCA, yes, exactly. So BRCA1 and 2, right? Those are common germline mutations. As we already heard from Joe, the mutations in P53, a gene called P53, That's a good example of a somatic mutation. So we do have an interaction of these mutations. Now, a good slide that Joe showed is the accumulation of those mutations over the course of time. So of course, we are familiar with the mutations that cause things to happen down the road, right? As a person ages. Great, thank you for an example from lung cancer, EGFR. So the concept that we're trying to build here is that notice that all of the things that you've mentioned here are names of genes, right? They are names of genes, and those mutations happen in those genes. Like, for example, BRCA1 and 2, those are BRCA1 gene, BRCA2 genes, right? EGFR is, again, a gene. So when we talk about mutations, we oftentimes talk about a change in a gene, and then we also will be looking at, well, what kind of change, right? So that type of a change could be insertion, deletion, a change, and different changes will lead to different changes in the protein that was encoded in that gene. So that's what we want to do today. Now, a lot of the material that you will find in a step-by-step overview is found in Genomics 1 that talks about, gives an example of a pipeline. So if you started looking at that pipeline, you will see that you will need 2 samples to identify a somatic mutation. And those 2 samples are going to be a tumor and a normal coming from the same patient. So typically what happens is there's a sample from the tumor, there's a sample from the blood of the same patient. You are assuming that you know that the tumor contains enough cells that are cancerous, and you know that the DNA is going to be the same in that blood sample. So by aligning that sample from the blood to the reference genome, you know that you are going to identify what kind of mutations, if you take the blood, the normal, and you align it to the reference genome, what kind of mutations are you expecting to find? Okay, the answer is germline. And if you align the ones from the tumor to the reference genome plus the changes, the germline, exactly, then you will find the somatic. So let's try and do that together. Let's go to the T-Bioinfo platform. We'll do it together. We'll be able to see how to do this using the demo pipeline first. And then as you go into the course, you'll be able to replicate the same using the samples that are provided inside the course. So if you go here to mutation variant, if you hover over here, this is analysis of exome sequencing data. So this is going to be data coming from exome-seq, so only the expressed regions of the DNA, about 20,000 genes. And you click right here on this drop arrow and click on the demo right here. So what sections do we have here? We have a section that deals with pre-processing. If you remember what Joe was saying, how the preparation of the library plays an essential role, what kinds of primers are being used, the PCR amplification, all of those things have an impact on the read quality. And so we can make sure that we are selecting only the reads that are of high quality by using this pre-processing step. Then we will, the first pipeline that we will do is actually we will map those processed reads onto the reference genome and visualize them. And that will give us a sense of what is in the data, right? So let's go ahead and do that. In fact, we can even skip the pre-processing right now. Let's just click on mapping, right? And visualization. This is going to be using a plugin called JBrowse. JBrowse allows us to visualize all of this data online, but of course you can also, if you have enough RAM and power on your computer, you can do this on your computer as well using integrated genomic viewer. So once you have completed this pipeline, you can see that I loaded the files. I map them on the reference genome, and then I visualize the alignment of reads. What kind of a data do I use for this pipeline? I use the exome-seq read file, which is going to be a FASTQ file. What is going to be mapped? What is the mapping file that I'm using? I'm using FASTA file, which is a file of the sequence of the human genome, and an annotation of that genome. So where are those positions of genes? That is going to be in the gene transfer format, GTF format. And then to visualize, I will have a SAM or a BAM file. So this is an alignment file, and that alignment file is going to provide me with the position for each read that has been aligned to the reference genome. So if those SAM and BAM files could be pretty large, so I'm not going to download this on my computer. Okay, another question, Samuran Jan. So essentially all of these do the exact same thing. They do mapping, star mapping, bow tie mapping, BWA mapping. Those are very commonly used. Okay, so yes, good questions. All these are good questions. So I have an alternative. Again, this is a demo. This is just first to illustrate one optional pipeline. Once you go into the Genomics 1 course, you will be given files to be able to create a pipeline. How do you know it's the correct pipeline? You have to understand the logic of the pipeline to be able to select the right function for the right logical step you have to do. So what are we doing here? We are trying to visualize reads. I think if you just walk through the steps that I'm doing, you will start understanding the logic of why we are aligning the reads to the reference genome and what can we see in the reference genome and what kind of information, biologically meaningful information, we can get out of this kind of a processing pipeline. So let's take a look, and then we will return to these excellent questions that you guys have. So first of all, I do start. Start basically loads the files. Then I go to mapping. I map on the reference genome. Right now, it doesn't matter which one I use. I'm just mapping. Let's go take a look at now the output of visually aligning those reads to the reference genome. Once I click on end, I have a button here that says visualization. I have it already open here. Okay, what do we see here? Let's just briefly review the interface. This is JBrowse. So JBrowse is an open source visualization of the genome. What do we see here? So first of all, you can see right here are the nucleotides, right? It goes from 0 to about 80 million. Right here, the next line shows you that we're actually looking at this little tiny area from this range. Okay, and where specifically are we? We are on chromosome 17. Right here, you can select a drop-down of all of the chromosomes. And right here, it says the position. So the position is 766414127692440. So we are looking at 28 kilobases. Kilobases means 1,000 bases. So we have 28,000 bases right here listed in this region right here. If we zoom in, if I just click to zoom in, you can see it says zoom in to see sequence. Let's just zoom in. Okay, now we are starting to see the actual sequence, and you can see those letters, which are letters, the nucleotides, and the codons that they code for. So now we understand that we are looking at a sequence of a gene. Let's zoom out a little bit, because this is the sequence. We don't need to see the full reference sequence. What we are interested to see is the gene. Now, what gene is this? This is a gene TP53. Obviously, this is P53. If you look on the internet for P53, you can, for example, find it on gene cards and read about this gene. And what you will find down here, if you scroll down, is the position of this gene on this assembly, which is the latest assembly, GRCH38, also called as HG38. So whenever you make your pipeline, you will actually be able to select this reference genome. So you can see that it's positioned on chromosome 17, right here in this area, right? And you can, of course, read about this. And why do we see here multiple versions of this gene? What do you think? Because look, there's black lines, red little lines, gray lines. What do you think all of this means? Variants. Okay, well, remember, yeah, different copies, different transcript. Great. So these are the correct answer is different transcripts or different coding sequences. Let's take a look at what are these different sequences. So first of all, the red areas are what's called exons. Exactly. Black ones are the introns. So we're looking at the introns and the exons, and we also see different copies of the same gene that have been found, right? So this is not related to the data that we loaded. This is just the reference. So you can see that there are multiple versions of this gene that are known, that have been found, with different exons that are either expressed or not expressed. And now this is all the information that we have about the reference genome in the GRCH38, right? So that is the reference genome. Now, where's our data? If we scroll down, now we can see this is our data, right? Because these are the two samples that we are looking at, sample one, or yeah, sample one, sample two right here. So these are different SRA archives. So those are archives of FASTQ files that you can find on NCBI. In fact, if you take this, oops. No, these are not from the 1000 Genome Project. This is from a project that has human patient-derived cell lines from breast cancer. So in fact, if you look up SRR 925765, SRR 925765, 925765, SRR 925765, right here. You will find that these are actually whole exome sequencing of a panel of breast cancer lines to identify mutations. So these are different types of samples taken from Array Express. I think they're also found on NCBI. But basically, they will have this pair. Why do we have two? Because we have a pair. One is a sample, and another one is a combined, is a pair of the same thing that is actually coming from the normal tissue, so from blood. Now, let's see over here, what do we see? Here you can see it says combined.bam, right? So this is the BAM file as a result of the alignment of the reads to the reference genome. We get a BAM file, and this shows us coverage and SNP, right? So this line right here shows us the coverage in terms of how many reads fall on this section. Now, if we look and we try to find which areas do those actually fall on, we see that the coverage falls on the exons. And that's how we know that this is whole exome sequencing. So this is sequencing of not the whole genome, because we don't see any of the introns. All of our reads simply fall on the exons. So this is a whole exome sequencing. Down here below, we see the alignment. Alignment of what? We see the alignment of reads. So let's zoom in a little bit more. Alignment of reads to those exons, right? So now you can see, how does this process actually happen? These short reads, right? If you click here, you see the reads, and this is the full sequence of that read. You can see its score. right? So that is what alignment does, is it produces you a score of alignment. It gives you the position. This is recorded in the BAM file. This is the position. This is the sequence and quality. You can see that each different letter has a different quality here. You can see that all kinds of information that the alignment does. For example, QC failed, no. So none of the quality control failed. And this is the actual sequence, right? So it's a very short sequence. It's about 100 nucleotide base pairs long. And now you can see how many of these do we have. What is duplicate? Sometimes you have duplicate reads. So if reads are many times duplicated, exact same sequence multiple times, those are typically the result of PCR amplification. Those are unnaturally appearing reads, and those are typically the ones that are being removed by PCR clean. Okay, now you also have blue and red. Blue and red are those, this is pair end sequencing. So for each read that comes from the beginning of, from one direction and the other direction are aligned, and that gives you higher reliability of the actual sequence, because as you remember, the sequencing itself also has a lot of error rates. There are other ones that are either not such good quality. So if you click on these, well, actually, yeah, they're not going to be, okay. So here you can see some of them have lower score. And you will also see that some of them will not have a matching paired end or a very short, like this, a fragment, right? So you can see, for example, this is 40, so about half of the normal size. So one of the challenges in alignment is not only finding the exact position of a read, but also analyzing how those reads match up to each other to produce a sequence that we can actually trust with a high confidence. Because now if you turn your attention to here, there are specific markings across this sequence, okay? And these actually show us the different variants that are found in this sequence. Now you can see that these variants will have different reliability scores for us, because some of the variants are only found in a single read, right? So here you can see those different variants that are found in just a single read, and those are essentially insignificant. Once they become multiplied in multiple reads aligned, we start having more confidence that this is a change. But nevertheless, here, for example, you only see 8 reads that contain this change, right? So if I see something happen 8 times from different reads aligned to the same place, I'm starting to think, okay, maybe there's something to it. However, look at my total alignment here. This is the y-axis shows us the height. This is the alignment. Exactly, this is the frequency that we need to now determine, and we will talk about that in a second. So if we go back and we kind of zoom back out, one of the questions is, what is an average alignment across the whole sequence that we are studying? So if we are trying to find a gene that has specific point mutations, one of the biggest questions is whether we have enough reads aligned to that region that will give us some kind of a high probability of seeing a real change. And you will see as you scroll through this visualization that some of them do and some of them don't, right? But this is just an example. What are the other steps, right? This is an intermediate step. It does not give us the answers to the questions that you're asking, right? The questions that you're asking, what should be the frequency? What should be the coverage? You know, which mutations to pick, which mutations not to pick. There's another question. Well, you know, this is just the cancer one, but also where are the germline ones? So now we can click on these and visualize them. And you will see that there's a whole different story for the germline mutations. And we're not going to wait for this to load, because now you can see, why did we talk about big data last time? Well, because this is a lot of information. This is just one of the genes, right? This is the region that we're looking, just on one of the chromosomes and not even the longest one. And there are so many different comparisons that we have to make to essentially be able to extract some kind of meaningful information from here. So what do we do with this intermediate step? Let's go back to the demo pipeline and create another one and see what are some next steps that have to be done for us to be able to process this data further, eliminate all of these unnatural duplicates, map the data onto the reference genome, and now we will introduce two other algorithms. One is called Strelka, which is a method for somatic single nucleotide variant detection and small indel detection from sequencing data of matched tumor normal samples. Okay, and now you can see what it does. Now you're familiar with what is a BAM file or a SAM file. You can also see that there's a normal and a tumor. You search for those indels, or so indels are insertions and deletions, right? You also have variants. So all of this is being, first of all, reduced for indels, insertions and deletions, and you somatic variants. And then you produce what a somatic variant file, VCF file. Okay, so that's what we want to do. Let's click on OK. We will also take a look at another approach that does something very similar. Again, looking at the germline genotyping algorithm. Okay, to find somatic mutations, let's click on End. And now you will see that we have a zip file, fairly large zip file. Let's wait for it to download. And I want to show you how to identify, how to see what's inside that file. Now, all of you are probably familiar with the integrative genomics viewer. I'm just going to paste this link right here. If you want more information on how to download and install this on your computer, all of this is reviewed in Genomics 1. So again, I'm just going to point your attention to lecture 2.3 in Genomics 1, in case somebody wants to go through all the steps to do this. You download the integrated genomic viewer from here, and I will just open it up on my computer right now. And now explain what we are looking for in this file. So here we are having a VCF file. So if you go back to the download, let's unpack this. Okay, we have here a VCF. Let's do file. Okay, I think it's opening. You will see, okay, there we go. You will see here that what I did was I opened this combined, right? So I can open, for example, each one of these files separately, or I can open them as a combined file. Let's take a look at this interface and see what's here. So here you can see that this is the reference human genome HG38, again, chromosome 17, and this is a place where you can search for TP53, for example. That is the position of the TP53 gene, right? That's the gene right here, TP53. Let's look for BRCA1. It's going to take some time to load. Again, we're on chromosome 17, but a different position. Okay, and now what do we see inside all of this information? Here we have, again, those coding and non-coding regions. But what we don't have is we don't have all of the reads. We don't have all the different variants that could be present in reads. What we have are the actual variants that have been determined by this variant calling algorithm. And we can look at it independently for each one of these files. We have 766, I believe, is the, okay, so yeah, let's forget about these actually. Remove track. Okay. And let's go with the aligned ones. So you can see here normal versus tumor. And if you see, yeah, you can see right here, this is the actual VCF. You can see where is this mutation found. If this mutation is found in both normal and tumor, that means that this is probably just a germline mutation. If it's found only in tumor, that means that it is a somatic mutation. So we can see that this patient specific, this cell line, has a couple of mutations in the BRCA1 gene in these positions. What do we have here? Here we have an assembly of mutations that are coming from the common SNPs database, right? So one of the next challenges, as you can see, the gene is fairly long. There are many possible mutations inside this gene. And for me to be able to see whether those have been reported on and added to some kind of a database, I need to have some reference. So one of the easiest ways to do it in IGV is when you go here, you can load from server, and it offers several different databases where you can look for annotation of the mutations that are found. Okay, we're just looking at common SNPs here. We have some other options here that we could look for. And you can also load some of the your own, or for example, external reference cases, which we will go through in later sessions when we talk about annotation. But the first illustration that we wanted to do here is that you can look at the raw data where there's a lot of questions that come in. How many reads is enough to detect confidence for detecting a single nucleotide variant? What are the different positions that we're looking for, which genes, right? A lot of questions. Or we can reduce this information and essentially go to a level of looking at just positions that we know are associated with something, right? And so here you can see that there's quite a number of germline mutations here. You keep zooming out, unless it gets stuck because it's too much data to show. You will start seeing eventually that there will be some tumor variants as well, but a lot more germline, right? And the thing is that this is actually expected. It is expected that there's a lot of interpersonal variation that is germline, but when we see those somatic mutations are typically cleared out by healthy normal cells. And those are repaired, or those cells need to die if those mutations pose a problem for the development of that tissue, for the development of that cell. So that's why we typically don't see that many mutations. And as you remember from our previous sessions, it actually does not take that many mutations in a single gene to start causing this abnormal growth and these characteristics of cancer, right? So let's go back to TP53, right? And you can see over here that again, we are seeing some variants, but those variants are only in the normal. Now, another problem here is that we're essentially looking at cell line data, right? And so what we will do in our future sessions, when we look at mutations, now that we understand how that process works, how we go from loading in a file of reads to then transforming it into an extracted set of data, we will understand what that actually is. Visual, we can visually, but essentially it's not an analysis that we can go to the next step very easily, because just to look through all of the genes that potentially I'm interested in, it's going to be very difficult. And we will go, for example, into a place where we can look at just a number of genes, like 5 or 10 genes that are mostly interesting to us, because we think that's where the most suspected mutations could be found. Well, that's easy to do here. But you will also see that there's a lot more information in the data that could lead to different conclusions. And our goal is to now understand the analysis steps so that we also understand how to deal with the output, not looking at it, not visually, but from a statistical perspective, selecting the most important and critical variants that we can then try to associate with a specific phenotype. And the phenotype here is going to be a subtype of a specific cancer, a specific prognosis, right? A risk factor, etc. Those are going to be the risk factors that we will look at. To learn more about all of this, we will actually turn to analysis of transcriptomic data in our next session, because in the transcriptomic session, we will actually understand how the different type of data in the FASTQ files and the different preparation techniques from next generation sequencing can help us analyze data that speaks about the actual process of genes that are being expressed. Okay, and Zainab here is asking if we can provide more resources. Well, that's exactly what we are trying to do in these sessions, but also, of course, we have the online material. If you are having trouble with accessing the online material, we're happy to send you those materials as a PDF, and then you can try practicing those on your own. But let me briefly conclude this session. So we talked today about analysis of genomic data, briefly saw how we have pre-processing steps, mapping and alignment, different options here, and we can read more about the different algorithms to understand how they differ. We talked about variant calling and the different ability to identify a change. What does that actually mean? Then differential variant calling, when we compare, for example, tumor versus normal, right? Somatic would be a differential, or we want to compare a bunch of samples with a bunch of other samples. And then we will talk about annotation. Yes, Zainab, we will definitely send you a PDF of Genomics 1. And then post-processing, right? What is the significance of those steps? We also looked today at one of the possible pipelines, and then one of the other sessions that we will have will be looking at statistical confidence and detection of frequency of mutations, which is going to be helpful. We talked today about we talked about differential variant callings, right? So for example, in the Genomics 1 course, you will read about uploading of the files and separating them into tumor versus normal, and then processing that data. And then today, what we talked about is, what is this visualization? How can we visually identify reads mapped to a specific region, and then find specific changes in that region? And discussed the role of P53. How do we find that gene? We need to know the location of that gene on which chromosome, right? And this is the location here. And we saw how we can find the same information but condensed that is going to reveal a change, whether it's somatic or germline, and how do we start interpreting that based on annotated known SNPs that have been found in other data sets. Eventually, we'll also get to learn about comparison of multiple samples. For example, if we have multiple samples from healthy individuals, multiple samples from cancer individuals, or we have different types, for example, some that are more aggressive or less aggressive. A question here, if a variant has an allele depth of 5 and the total read depth is 20, so the variant read frequency will be 0.25 or 1/4. Is it significant to interpret? Well, your question is about the significance for what, right? That's the other question here that you're not considering, because if you are thinking of whether one mutation is significant, for example, to risk, for a person at risk with cancer, or your question is whether it's significant to say that that mutation makes this tumor more aggressive or less aggressive, right? That's the other question, is the question of the phenotype. And then it's not only, well, for annotation, of course it is significant, right? For annotation, first of all, do not just consider the depth, let's say of 20 in this particular region. You're also considering, well, how is it distributed across the whole exome? And that has to do with just the total coverage for your file. So those are some technical questions that are very good, but I think you also need to consider the larger context of what is the sample and what are we talking about? A gene that has 1,000 nucleotides, that has 20,000 nucleotides, how many nucleotides does it have, right? What is the probability of finding a single variant in that length of a gene? And then looking indeed at depth, but not only a depth in that particular point, but also depth across that gene or across that region that you're looking at. And that's why there's a complexity to these algorithms that essentially produce the mutations that are ready for annotation, right? The VCF calling, the calling of those variants, produces a file that says, out of all the possible positions that we've analyzed and we saw a change in, these are the ones that are good for annotation and interpretation, right? That's the point of those algorithms. What goes into it? Many different steps of first preparing the reads, then making sure that those reads are aligned to the best position, then making sure that you have enough reads out of how many you have per region, per gene, etc. And then a conclusion of something that you would like to annotate in the context of one single sample, or as shown here, in the context of a comparison between different regions. Exactly. Coverage is very important. And coverage is relative because typically when you compare several different samples, they might be done with the same coverage, they might be done with different coverage, right? And if you look at COSMIC, for example, another question is, well, when you put something into a database for annotation, how many samples is it based on? You will find in COSMIC, for example, they have hundreds of thousands of samples to be able to add something to a database and really start thinking about the significance of that variant for a phenotype, right? Now, I do encourage some of you to take a look at this overview of somatic mutations in cancer and normal cells. I'm just to paste it here. There's actually a few links that are available that I think everybody could benefit from based on the questions that you might have. Okay, here's a couple more links. Now that you understand a lot of these concepts practically and visually and can, oh, sorry, I'm looking at everyone here. Those are some of the links. Please consider looking at those briefly. Okay, so we're already a little bit over our time, but if anyone has any questions, I'll be happy to answer those now. Okay, because we're running out of time and we have another session here coming up soon, I want to mention some of the things that we'll be talking about next time. Okay, let me pause with that. Do we have to have normal sample always? Well, if you think of a somatic mutation is what you have based on the germline mutations that have already been in the genome of that patient, right? Because the patient has been, you know, we are using a reference genome. That reference genome is based on, you know, multiple different sources of data. So an individual is going to already be different from the reference genome. And if you want to only look at somatic mutations, which are going to be accumulating over time, then you want to exclude the germline mutations. However, sometimes you want to just study the germline mutations, right, which also contribute to the risk of developing cancer and might have a specific interplay with treatment or with how fast the cancer progresses. So I think it's not that you need to have normal sample always, but it's definitely very helpful to have that depending on the question that you're trying to ask. And yes, we will send the links by email as well. So next time we'll start talking about transcriptomic data, right? The same kind of FASTQ files that we looked at for variants, we will be talking about in terms of expression. And we also have a number of online courses that you can start looking at, and Zainab or anyone that's having internet issues in your home country will be happy to send you those PDFs so that you can take a look at those. And then what we'll do is we will start learning about some projects that illustrate some of these materials in the context of a specific question and a specific data set, and not just theoretical, right? So our goal is to move slowly up until we get to a point where we can clearly look at the data, clearly look at the analysis, and then to start talking about specific projects. Again, thank you for joining today. I hope that this session was helpful, and we're getting closer to where you want to start applying these methods to a data set. And we hopefully will see you again next time. Thank you.